The Sainsbury Laboratory in Norwich works in close collaboration with numerous research groups locally and across the world. This broad network of collaborators and its large number of visiting scientists strengthen the laboratory as a scientific hub. Founded in 1988 with just 22 employees, driven by dedicated scientists, the lab has grown to position itself at the cutting edge of plant microbe research.
What's been really fantastic about being at the Sainsbury Lab nearly 25 years is to be part of an institution that's made such an incredible impact worldwide, starting from really ground zero, starting from nothing. Of course, there's the RNAi, there's the cloning of a lot of resistance genes, and now we've transitioned to doing genomics on the pathogens, to understanding what all the proteins, or at least many of the proteins involved in the interactions are doing. And we just keep peeling back more layers of the interaction. And every time we peel back a layer, we see new things that provide interesting insights and provide new opportunities for intervention to elevate disease resistance in crops. So you can see this whole chain from starting from essentially nothing to a huge raft of discoveries, and then you can see the great potential for making a big difference in the field.
More than 2,000 scientists from 25 nations have been employed at the laboratory over its 25-year history. Today, around 85 scientists from 25 countries work at the Sainsbury Laboratory, forming a unique, multinational community of researchers driving forward outstanding research.
I'm very glad to be here because all the colleagues and the friends around, they are coming from all over the world, and we really enjoyed the pure science here, which we explore a lot of new things every day. I really enjoy it.
And the culture and everything, it's just fantastic with the international culture that we have here.
At the Sainsbury Laboratory, everyone is asking the same questions. How do plants sense potentially pathogenic microbes and acquire immunity? And how do pathogens overcome immunity and cause disease?
There's a lot of work getting done in the same general area, which is nice because then you can discuss things with other people.
When do pathogens cause disease and why? It is in this field that TSL has established itself as a world leader.
One of the best places to study plant science, and it was imagined and enabled by people who love science, and then it's managed by scientists with a lot of degree of freedom to do their research. I feel really fortunate that I was part of this adventure, and first as a postdoc and now as a group leader, and I'm really looking forward for the future to carry on this great adventure.
For their research, the scientists work with various model and non-model organisms. These include Arabidopsis, tobacco, tomato, potato, wheat relatives, and soybean. The pathogens include bacterial pathogens, rust fungi, and 3 types of oomycete: Phytophthora infestans, Hyaloperonospora arabidopsidis, and albugo. Using the most modern technical methods, TSL scientists have been able to determine the genomes, transcriptomes, proteomes, and screen phenotypes from the whole organism right down to the subcellular scale. This data has helped solve some of biology's fundamental questions.
What is fantastic about TSL is the possibility to drive research to new areas, and this is greatly supported by big investment into technologies. And in fact, although this is a relatively small institution, this one was one of the first places getting a next generation sequencing instrument. And when I started my research here, the lab committed to buy a high throughput confocal microscopy platform, and these are only 2 examples that demonstrate that technology is driving research, but also the research is pulling that technology in, and in order to have both things working together, it needs an excellent bunch of people.
The scientists at TSL never restrict themselves to specific processes in plants. They also want to know how the pathogens interact with these processes. This non-restrictive approach produces huge datasets, which are evaluated by dedicated bioinformaticians, together with the researchers at the laboratory.
The best thing about working in the Sainsbury lab is that I get to work quite closely with the bench biologists and the other scientists, and that helps me discover things that nobody has ever discovered any time in the world ever. That's a really cool thing.
Scientists at the laboratory perform fundamental research. However, many of their results can be applied. With the help of combined genomic and functional screening approaches, novel leads for resistant crops are being examined. Here, the Sainsbury laboratory partners with the Two Blades Foundation.
In recent years, TSL has taken a growing interest in turning its basic discoveries into practical outcomes. We call this the TSL Plus programme.
The Sainsbury laboratory's research success is formed through support from the technology teams. Bioinformatics, proteomics, transformation, and the newly formed SynBio support team, and in partnership with the John Innes Centre.
When I started here, I was blown away with the support that I can get. So the only limiting factor here is my imagination.
The scientists conduct their research on equal footing with flat hierarchies. Interactive networks promoting collaboration are a big part of TSL's successes, and these are fostered by annual retreats, regular seminars, and discussions, tightening the bond of this multinational scientific community.
We have lots of interaction with lots of different people, really lots of interaction between the different groups.
Over 25 years, TSL has attracted highly talented scientists at all levels to analyse the processes of the interaction between plants and microbes. The laboratory provides excellent training opportunities to today's 65 doctoral students and postdoctoral researchers, helping nurture young researchers into the world-leading scientists of tomorrow.
The lab attracts and nurtures the best. The quality and intellect of my colleagues, from the scientists to the support staff, is just phenomenal. As a result, there is a distinct buzz about the lab, the sort of vibes one gets in happening places.
Ultimately, the Sainsbury laboratory funds people rather than projects, and 6 monthly meetings with the International Scientific Council ensure the direction and excellence of the research.
We come every 6 months as council members and we see exciting science, and it's a real privilege to have been able to observe the Sainsbury people for the last 6 years and the really exciting science that they're doing. I listen to these people describing projects that are their most creative ideas because they have a source of support that doesn't depend on the project, and so they're able to pursue their most creative ideas. It's about the people, it's about the quality of the people, yes. Right? So there's a unique group of individuals here that have been selected among the very best. So kind of the best of the best.
The excellence of the community, the freedom to perform the most creative research, and the unique culture at the Sainsbury laboratory will ensure the continuous achievement of its primary goals, to gain knowledge and to provide solutions for agriculture. Science is a lifestyle at the Sainsbury laboratory.
But,
Well, I'm going to get giggles now.
What's so great about the Sainsbury lab is the quality of the colleagues we have. Come on.
The best thing about TSL is the TSL football team.
Okay.
I mean, it's always a challenging job here. You keep up with all these new things, interactions with people. But one of the biggest challenges, of course, is learning how to ignore Roger Friedman, who swings by from time to time. And after 25 years, I've started to get pretty good at it.